Okay, hi everyone. Happy Friday. So we have today's lecture and Monday's lecture will be the last two lectures of new material for the semester. And so I think we're wrapping up our semester talking about the role that the immune system plays in preventing cancer. And I think this is a wonderful way of wrapping up the semester because it will hopefully put together a lot of the things, a lot of the concepts that we've been talking about over the course of the course, and actually apply it towards understanding how the immune system has a very important role in actually maintaining the normal health and homeostasis or proper physiological balance and behavior of our normal tissues. When we think about the immune system, I'm sure most of you at the start of the semester, before we started learning about how our immune system works, if you were just asked by somebody, hey, what does our immune system do? I think you probably would have said, oh, it protects us against, germs, pathogens, right? Like bacteria and viruses, things that we've certainly learned about. Over the course of the semester, hopefully you've come to appreciate that the immune system actually does much more than that. Protecting us against pathogens and infections from pathogens, of course, is a very important component, an important job of what our immune system does. But really, an equally important and perhaps even more important job is protecting our bodies against our own cells that are going bad, right? Cells that are changing and potentially could be problematic or threatening to our health. So cancer is a discussion of that. And so we'll talk today about what cancer is so that everybody is sort of on the same page with understanding what are some of the fundamental sort of concepts in understanding the development of cancer and why then cancer presents certain types of signals for our immune system. And we'll look then at how our immune system is able to monitor our tissues, constantly searching for cancer cells to eliminate before they can cause problems and actually cause cancer as a disease in our bodies. And then on Monday, we'll wrap up by continuing our discussion, but we'll look at different ways that our immune system can be leveraged therapeutically to fight cancer. Okay, so what is cancer? Cancer, I'm sure all of you have your own concept of what cancer is. Many of those concepts are probably based on very personal experiences, right? People that you know, friends, family who have been impacted by cancer. Right? But what is it? Very simply, cancer is a loss of control over cell proliferation. In the most basic sense, when the cells in your body lose the ability to manage how often they divide, and if they begin to divide out of control, right, that is the start of potentially turning into cancer. So let's back up a moment and let's review some basic cell biology to make sure that we're all sort of on the same page here. Right? Recall that every single nucleated cell in your body, right, every cell in your body that has a nucleus, contains in that nucleus a complete copy of the human genetic blueprint, right? The human genome, your human genome that you inherited, partially from mom, partially from dad, right? And that is your genetic blueprint. So within that blueprint, our 23 pairs of chromosomes, we have something on the order of 20 to 25,000 different genes. These genes code for different proteins, proteins that perform all kinds of functions, both structural proteins, as well as proteins that function as little machines that carry out molecular tasks. So these would be enzymes, for example. And I should say a third group of proteins that act as signaling molecules, right? This semester in immunology, we learned a lot about signaling molecules, those little proteins that we call cytokines that serve as messages between cells of our immune system, and how those cytokines are a critical part of whether different immune cells can communicate with each other. So of course, those cytokines, because they're small proteins, they're coded for by the genes that have the right genetic sequence to result in the expression of the properly formed protein. And that protein will then function as a little message because it can be received by other proteins like receptors. Right? So if you're talking about any given cell in your body, we're talking about normal, healthy cells that make up your different tissues. What makes a skin cell different than a nerve cell or a muscle cell or a hair follicle cell or a gland cell somewhere in your mucosal layer, right? It's not that they have different genomes. They all have your genome, right? It's just that different cells use different subsets of those 20 to 25,000 genes, right? A nerve cell will only express those set of genes that it needs to be a nerve cell, right? Compared to a muscle cell, which may express many of those same genes, right, that are used by all cells, but then among those would be a sort of a separate subset of genes that are more specific for a muscle cell to function as a muscle cell. Consider also that the whole process of the cell cycle, as the cell undergoes replication of the genome heading towards mitosis, right, eventually the dividing of a parental cell into two daughter cells, each with a perfect copy of the starting genome, that whole process itself is controlled by many proteins working together. Right? And those proteins are coded for by their own genes. So therefore, it's very correct to say that genes also control when a cell should divide and when it should not divide. So that's why in basic cell biology, we are taught that a given cell in your body, if you were to randomly pick a fully differentiated cell in your body and you were to sort of see what it's doing, that cell is either going to be in the process of dividing or it's going to be in the process of doing whatever its assigned job is, right? But importantly, a cell cannot do both things at the same time. Most of the cells in your body that you see when you look at yourself in the mirror are what we call fully differentiated cells, right? These are cells that originated as stem cells, found in the tissue that divide and give rise to different cells that gradually turn on or turn off certain programs or subsets, since that's the word that I used already, subsets of genes in order to become, right, the correct type of cell that they're meant to. And so the majority of our cells in our body are differentiated and they're just doing the job that they're assigned to do. So at any given time, the majority of cells in your body are not actively dividing. So that term differentiated implies that at a certain point, right, the cells that make up the sort of final type of cells that are going to work in your body, right, they've reached a point where they're probably no longer going to be dividing anymore. So that means the pathways, the different signaling pathways of different proteins inside the cell that control things like cell proliferation, right? Those may not be the pathways that are being emphasized or used if a cell is just performing its normal cell function. So how many cells are there in the human body? Those of you who have taken cell biology from me before, have probably heard me talk about the idea of we have not just billions of cells, but trillions of cells, right? And once you get up to the point of talking about trillions of something, I don't think in our everyday lives that's a number or a scale that we're used to thinking about. I mean, we can think about things like hundreds of something, right? We can think about, oh, I'm pretty comfortable with an idea of what 1,000 of something is, right? Or even, 10,000 of something. But once you get up to like millions of something, billions of something, right? That's no longer practical. We don't really think in everyday terms of things that that get that high in number. And so certainly once you get to trillions, it's almost like, I don't even know what that is, right? It might as well be a made up number, right? But indeed, our cells have trillions upon trillions of our bodies have trillions and trillions of cells. So let's think about this on a scale that we might be able to appreciate easier. Imagine you're walking down the sidewalk, right? And you find a dollar bill lying on the ground, right? So there you go. By the way, these are not my feet, right? In case you're wondering about, like, those are not my shoes. Okay. But we've all been lucky enough at some point, right, to maybe find a dollar bill lying on the sidewalk, right? So you have a sense of the size of a dollar bill and what that would kind of look like sitting in front of you, right? So let's imagine that's one single dollar bill. What would a trillion dollar bills look like? So imagine you're standing there looking down at your feet. Now imagine a trillion dollar bills, right? So here you are with that $1 bill at your feet, and then these are stacks and stacks of single dollar bills, right, piled up into pallets. And so 1 trillion individual dollar bills would look something like this. Right? I think this illustration was actually meant to originally demonstrate something about like the US debt or something like that, right? So that's why there's like the White House and other government buildings in here. But that is 1 trillion, right? That's a huge number to deal with. So how many cells are actually in our body? Way more than that. The human body has around 11 trillion nucleated cells, right? And so that's not even counting your red blood cells. Remember, your fully differentiated red blood cells that are circulating around in your arteries and veins right now, right? Those cells, as part of fully differentiating and becoming functional mature red blood cells, have lost their nucleus. Right? So evolutionarily, our red blood cells have gotten rid of their nucleus so that there's more room inside of the cell to carry things like hemoglobin. Right? So red blood cells, we have about 22 trillion red blood cells. Right? So that's an interesting fact, right? That if you were to say, what's the most common cell in the human body? The answer would be, oh, the fully differentiated red blood cells in our blood. And we have about 22 trillion of those. And all the other cells in our body, if we think of as the nucleated cells, we still have 11 trillion of those. Right? So of those 11 trillion, each cell then, right, has its own nucleus, and that means it has its own set of genes that control things like whether it should divide or shouldn't. So cancer then arises when one of those cells starts making mistakes. Right? The control of those genes and the resulting control over whether it should divide, right, is compromised. So that's kind of a scary notion. If we're talking about 11 trillion cells that make up you, and obviously that number kind of varies, right, based upon your body size, etc., but roughly 11 trillion cells right now, right, that are part of you, and each one of those cells has the potential to lose control of its proliferation and start forming cancer. Right? So is that scary? It is scary. It's kind of terrifying in a way, but you have to have some faith in the fact that, wait a minute, right? If we didn't have a way of controlling such cells, detecting them and getting rid of them, then let's face it, you wouldn't even be sitting there in the first place, right? So the fact that we are sitting here today means our body must have really good ways of managing the detection and elimination of these cells that are turning cancerous. Every day in your body, if you're talking about 11 trillion cells that potentially can turn cancerous, you better believe, right, there's more than just a handful of them that are starting down the pathway to cancer. Right? In fact, just by your typical everyday being around things like walking from class to class on campus. I know we don't do that anymore, right? But just walking around outside, being exposed to UV radiation, just from eating different types of foods, right? You have literally, right, millions of cells in your body that when you woke up this morning were perfectly fine, but by the time you go to bed tonight, there are mistakes in the genomes of those cells due to different exposures. Right? And so by the time you go to bed tonight, you have millions of cells in your body that are starting down the pathway to eventually becoming possibly cancer if they're not stopped. Right? So we have really good mechanisms for making sure that doesn't happen. Let's talk about some cancer terminology here. So, of course, we're talking about mutations. I think all of you know that cancer is based upon mutations, right? Alterations in the genetic sequence of those genes that somehow play a role in the control over the cell cycle and whether the cell wants to divide. So mutations in genes, they may be silent mutations, meaning just because you accidentally change the identity of a particular nucleotide from, let's say, a T to a C, right? Maybe the place where that is on the long string of DNA, that's your chromosome. Maybe it doesn't really matter what that nucleotide is, right? Because maybe it's not part of a gene, maybe it's someplace that doesn't even serve any sort of important regulatory function. Right? Sometimes, right, a mutation might actually happen in the middle of the coding sequence of a gene, but again, it might be silent in the sense that it might be that it changes the identity of the third position of a nucleotide codon, all right, that codes for an amino acid. And you remember, again, from basic cell biology, that our genetic code is set up as triplicate codons, right? Every 3 bases in our DNA, ultimately, when they are transcribed into RNA, code for one of 20 amino acids to be put in that location in the growing polypeptide strand that the ribosome is assembling as it is translating that RNA. So that third position of the codon is the wobble position. It may be you get a mutation, but it ends up not changing the identity of the amino acid that ultimately gets put into that spot of the growing protein chain, right? So in which case that could be silent as well. However, when we think about cancer, what we're talking about are mutations that do have a dramatic effect that might lead to either loss of proper protein function. So when the protein is made, because of this change in a particular DNA base, an incorrect amino acid is put there by the ribosome, or it might end up causing the ribosome to prematurely truncate, right, or stop translation, so you only get part of the protein made. Right? Even if you get the whole protein made, maybe that one change in amino acid changes the shape of the protein ever so slightly, right? But as I think all of you know, the proper functioning of proteins is all dependent upon 3-dimensional shape and how they're folded up. So even a slight alteration might lead to sort of a loss of that protein's proper function. Or the other side might be that a change in the shape might actually increase the activity of the protein improperly, make it work better than the cells of your body are expecting it to work. Right? So that's why it could be a loss of function mutation or a gain of function mutation. Either one can contribute to the loss of proper control over the cell cycle if the protein that we're talking about is one of the many proteins involved in making such decisions. Right? So when cells start dividing out of control, this leads to tumors, right? So tumors are just sets of cells that are growing out of control. And these may be solid tissue tumors, or they might be liquid tumors, right? I know liquid tumor sounds really disgusting, but I mean, that's what hematopoietic cancers are. Right? Things like, for example, B cells or T cells, we'll just say the lymphocytes. Right? As lymphocytes are developing from precursor cells, if control over division is lost, then you might have tremendous numbers of improperly formed lymphocytes floating through your blood. And that would be like a liquid tumor, right? As your blood is just filled up with these improper, malformed lymphocytes. So the tumors themselves can either be benign, which we would say is non-cancerous. So just because you have a tumor, that does not mean you have cancer. That's a very important distinction. For example, it is quite common in people as you get older to have things like polyps, right, in the lining of your, let's say, your colon, right, your large intestine. And these polyps are small little tumors, but it does not necessarily mean that they are disease, right? These are just cells that have lost a bit of control over division, but they're still somewhat limited there, right? A lot of times, if you have certain types of, things on your skin, for example, right, little bumps, things like that, those can be considered tumors where skin cells are dividing a bit out of control more than they should, right? But you certainly don't think of that as necessarily cancer. However, right, oftentimes when you see such an occurrence or such a growth, right, you're encouraged to go to the doctor so that they can keep an eye on it to make sure that it really stays benign and it doesn't show the hallmarks of actually becoming cancerous. So when something becomes cancerous, we say it becomes malignant. Right? And this is when tumors gain through continuously messing up and getting more mutations. Right? Once they get a certain number of mutations, that gives these cells additional properties. Right? It changes their behavior, and some of these changes in behavior might actually contribute to the aggressive nature of them dividing even faster or potentially spreading beyond the normal boundaries or limits of the tissue that they started from. Right? If that happens, we say they becomes malignant, then you're talking about the start of cancer. And if those cancer cells manage to leave the original spot where they started from and spread to other locations in the body, that is what we call metastases. So let's just look at this cartoon here. So here we have on the left example of a benign tumor. So this is an adenoma, meaning that these are some gland cells somewhere in one of the different glands in the tissues that have begun to lose control over proliferation. So the whole sort of background pinkish sort of brown field here is the tissue, and you're looking at a cross section of the tissue, and in this tissue you see lots of ducts, right, little vessels flowing through it that are sort of caught in cross section, right? So each one of these would be the normal cross section of a little duct, and the darker brown sort of inner lining of that would represent all these normal epithelial cells that make up the inner wall of the duct. Right? And then the black line around them would sort of represent a membrane, a basement membrane that separates those cells from the surrounding normal tissue. So what's happened in the middle here? What has happened here is somewhere along the line, one of these individual cells that are part of one of these ducts, right? So keep in mind the dark brown, actually, this is not one single cell. Right? This is a cross section of a duct that's made up of many cells. So the darker brown thing, right, inside is a whole bunch of these normal ductal cells on the inner wall, right? So at some point, one of these cells somewhere in here began to lose control of dividing. And over time, it's lost control of proliferation. So now you have a bunch of these sort of, densely packed ducts here, right? And this is the start of a tumor. Right? Importantly, this tumor is still surrounded by the boundary that separates it from the rest of the surrounding tissue. In this case, the cartoon is saying, okay, you imagine there's like a fibrous connective tissue capsule of this benign tumor, and you can almost imagine the cells inside kind of pushing up against it. Right? But as long as this condition stays this way, in other words, if whatever mutations these cells got, right, that make them proliferate a little bit more rapidly than normal, if in reality they're sort of kept in check, because as they proliferate more, they're also dying off, right? So the tumor doesn't really get any bigger. It's confined within the space. It kind of stays there. Then you might say, yeah, this is a benign tumor. Maybe it's not worth it to go in and operate, right? Because depending on where this is, it could be pretty deep inside of the body. It might potentially be more risky to operate and open a person up to try to get to this and take it out, right? It might be more risky to do that than to just accept the fact that, this benign tumor doesn't look like it's going anywhere fast. Let's just watch it, right? So every time you go to your doctor, they do a follow-up and they see, is it changing at all? On the right is what happens if something like a benign tumor eventually starts getting more and more mutations that mess up more and more control over the cells, right? And eventually lead to things like them breaking out from the original tissue location that confined them. So in this case, the red ones here are these now cancerous cells or cancerous tubules, right, that are made up of all these cancerous red cells. Okay. And one hallmark here is, of course, you can see that they are very messy shaped, right? They no longer look neat and tidy as these cells are becoming progressively more messed up. All right. So it's important to realize that all cancer starts from one normal cell, right, somewhere in the body. Okay, so cancer is not something like an alien that comes from outside and suddenly sets up a new home inside of the tissues of your body. No, right? I mean, I think you guys know that, but I also think you'd be very surprised. I think a lot of times in the general public, people are so scared of cancer, right? It's the big C, right? That everybody's so scared of, but they don't really understand what cancer is. So all cancers begin with a single normal cell in the body that, because of getting the right combination of a couple of mutations, begins to transform. That's the term that we use, right? And eventually this transformation, if it's allowed to keep going and additional mutations are added on, right, to those existing mutations that are being carried through by these cells as they're dividing, it's possible that this transformation can lead to becoming cancer. So not surprisingly, some mutations in some genes encourage this process more than other genes, right? So there are two most important categories of cancer-associated genes that we can talk about. These would be certain genes that are normally involved in encouraging a cell to want to undergo mitosis. So importantly, all of our cells have these genes, right? Because in order for a cell to even do normal mitosis, there must be these genes that drive it towards that. So those genes that do encourage a cell to go through the cell cycle towards mitosis, we call them proto-oncogenes. Right? If one of those genes gets a mutation that changes its function, for example, it gives it a gain of function mutation. Now suddenly these proto-oncogenes, or I should more specifically say the proteins that are made from these proto-oncogenes, no longer behave well, right? They start behaving out of control. Then they are what we call oncogenes. Right? So a proto-oncogene is the normal precursor of an oncogene, and the only difference is the proto-oncogene form of it is the normal gene that all of your cells have and rely on, right? But if it's mutated in a certain way and it loses proper behavior, right, so that the protein that it makes now is no longer controllable, and as a result, the cell starts getting pushed more and more to divide, then we say that mutant form of the gene is an oncogene. So a couple examples of oncogenes are things like MYC or RAS. So those of you who do any kind of reading about cancer research, oftentimes oncogenes like MYC and RAS are implicated in particular types of cancer transformations. On the other side, the flip side, we must also have genes whose proteins that are made from them function to kind of put the brakes on improper cell cycle progression, right? So these would be what we would call tumor suppressor genes. They halt cell division. So examples of these TSGs or tumor suppressor genes are genes like P53 or PTEN. So by presenting it this way as these two groups of genes that are sort of diametrically, engaged in kind of this tug of war, right? Certain genes, their proteins function to drive the cell cycle forward. Other genes, their proteins, right, function to halt the cell cycle. It is very much like the two pedals of a car, right? Your accelerator pedal, the gas pedal, and your brake pedal. These gene groups work against each other to determine whether the cell moves forward with division or stops. In my cancer class, I often use this analogy of the cell being a two-footed driver. Two-footed driver means I think most of us, we drive with one foot, right? And we actually alternate whether our foot's on the gas or on the pedal, right? There are some people who, I guess are more comfortable driving with 2 feet, one always on the brake and one always on the pedal. And in some extreme cases, they're always sort of pressing both down, right, in this almost very sort of cautious way. Occasionally, you'll see somebody like this on the freeway if you're behind them because their brake lights are always on, right, even though they're driving forward. And I think you understand what's happening. Depending upon which pedal gets pushed harder at a given time, right, that influences whether the car moves forward or slows down and stops. So in that same way, these two groups of important cancer-related genes, the oncogenes and tumor suppressor genes, are just like that. And whichever side sort of wins out, that ultimately decides what happens to the cell. Throughout this semester, we've discussed this concept of how cells are constantly receiving messages from the outside, right? That might be stimulatory and some of them might be inhibitory. And it's all about the balance of which signals are stronger at a given time that end up driving the cell, for example, a cytotoxic natural killer cell or T cell, right? Ultimately, it's that which side wins out in this tug of war that ends up causing that cell to activate and maybe kill a target cell. So if you understand that in terms of external signals, those external signals are all triggering internal pathways inside the cell, right? So it makes sense that inside the cell itself, you have constantly these types of pathways going on that are always in a tug of war, controlling things like cell cycle. An important thing to keep in mind is on any given day, when you're out there driving your car on the freeway, like on H1, for example, and you're surrounded by hundreds of other drivers, let's face it, we may think that we ourselves are pretty good drivers, right? But you all know what it's like to notice somebody in the car next to you who's not paying attention, texting while on the freeway going 70, right? Or, clearly distracted, right? But what can you do? Every day when we drive on a busy freeway and we're surrounded by other drivers, we have to have some sort of faith in the fact that the other drivers around us know what they're doing, right? I mean, yeah, it's good to be a defensive driver and be aware so that when somebody accidentally cuts you off because they didn't look in their blind spot, you can react, right? But we can't always just be scared that the cars around us don't know what they're doing, right? So that's just like the cells in our body. There's no way that we can actually control what every single cell, the trillions and trillions of cells in our body, are doing. So your body actually has to have some faith in the fact that the individual cells themselves, the individual drivers of all the cars on the freeway, know what they're doing, right? And should have some responsibility about their own driving behavior, just in the same way that the individual cell in our body takes responsibility over its own proliferative behavior. So many tissue types give rise to cancer. I already said that any nucleated cell in your body, any cell that has a nucleus, has the potential to transform and become cancer. So it's not surprising then that there are many, many different types of cancer. Right? So here's just a overview, just a general example. So cells of your lymph glands can give rise to lymphoma. Cells of your lungs can give rise to lung cancer, pancreatic cancer, bladder cancer. The hematopoietic cells of your bone marrow can give rise to hematopoietic cancers like leukemia. Skin cancer, of course, here in Hawaii, we're all very aware of skin cancer from UV damage from the sun. Breast cancer, kidney, colon, various cancers of the urogenital tract as well, in the reproductive system, right? So I think this contributes to the public's sort of vague perception of cancer, right? And the general public doesn't really have a good appreciation for the fact that because you're dealing with so many different cell types in the body, right, that means different cells go bad or go rogue in different ways because they're using certain sets of genes that they depend upon to be the normal type of cell that they're supposed to be. So this tissue variability presents a unique challenge for treatment, right? But again, I want to emphasize here, every cancer begins with a single cell undergoing transformation. So let's kind of bring immunology into this now, right? This may or may not surprise you, but many cancers are caused by viral infection. Because after all, if cancer is all based upon improper alterations of your genetic sequence in your DNA, right, mutations, then viruses certainly do that. Many viruses, they infect by releasing their genetic material into your cells, and then their genetic material becomes incorporated randomly into your genetic material, right? That's how the virus hijacks your cell so that your cell is now making proteins based upon the viral sequence. Because of the random nature, often, of the way the viral genomes integrate into our DNA, that means there can be all kinds of random disruptions in things like our genes, right? Our normal human genes can be disrupted by having pieces of viral genome put in them. After all, DNA is DNA, right? Whether it's DNA from a virus or DNA from a person, it's still just a molecule that's a series of A's, T's, C's, and G's, right? So around 20 to 30% of cancer types, now I stress this, right? I'm talking about roughly a third of all cancer types, not frequency, are linked to viral infection. What I mean is this, right? I'm not saying that if you take everybody out there who has cancer right now, 1/3 of them was somehow their cancer was caused by virus. No, that's not what I'm saying. What I'm saying is if you make a list of all the different types of cancer, lung cancer, bladder cancer, breast cancer, skin cancer, right? You would find that about 20 to 30% of that list, those examples have some sort of link to viral infection. Here's just some examples here. As is pretty typical with tables like this in this class, I don't expect you to memorize this, right? Because I don't want you to spend your energy trying to memorize things. This is just to give you an appreciation of just some examples of links between viruses and cancers, right? So certain DNA viruses, like papillomaviruses, give rise to tumors, for example, cervical cancer caused by HPV, right? Human papillomavirus. Hepatitis B virus gives rise to hepatocellular carcinoma or liver cancer. Epstein-Barr virus can give rise to different lymphomas. Then you have the viruses that carry their genomes as RNA, not DNA, right? So things like, well, HIV, right? We studied HIV. HIV carries its genome as RNA, right? And we already learned that HIV infection can lead to AIDS, an immunocompromised condition, where then certain cancers like Kaposi sarcoma, right, that normally a healthy, robust immune system would prevent from happening, in somebody who has full-blown AIDS and they're not able to mount a good, strong adaptive immune response, then certain types of opportunistic diseases take over, including forms of cancer that are normally not an issue in healthy people. Right. So how does our immune system actually get involved here in detecting cancer cells? A single cell in our body that is transforming because of random mutations and cannot stop itself from dividing should, right, be able to, on its own, realize that it's in trouble and on its own undergo apoptosis. So in other words, our first line of defense against cancer are our cells themselves doing the right thing if they start transforming and just eliminating themselves. That's what I was saying with the whole freeway driver analogy, right? You sort of have to just trust that the other drivers around you on the freeway, as you're all going 70 miles an hour, know what they're doing, right? So if, though, certain mutations that are part of this cell transforming happen to be mutations that affect its ability to realize that it's going bad and or be able to carry out apoptosis, right? Because after all, those are all a function of genes that code for proteins that allow it to do apoptosis properly. What if those genes have mutations and it cannot do apoptosis? Those cells then will persist, and that is your risk of cancer. Right? So that's why our immune system is our second line of defense. Our first line of defense is trusting that our cells are equipped to detect themselves if they have problems they can't fix and eliminate themselves. If they fail to do that and they keep dividing, then our immune system steps in as our second line of defense. So I want you to consider again, our immune system protects us from external pathogenic threats. And now I want you to think about all the internal threats that are coming from our own defective cells. And if I've scared you before by saying that in the course of waking up today to going to sleep tonight, you will have millions of cells in your body that are starting to transform through mutations, right? Hopefully you realize that the vast majority of them themselves will just undergo apoptosis and take themselves out of the picture. But those that don't, right, our immune system protects us 24/7 from those cells. So such immune responses are just like attacking foreign antigens in many ways. So here's this cartoon here. So in this kingdom, we have this the appearance, right, of this monster, which we'll say is like the cancer cell that came from within. Right? And so the heroic knights that are going to protect the kingdom against this cancer cell are very much cells like our CD8 cytotoxic T cells that are attacking a cancer cell, right, as we see here. Right, so these pink things are our cytotoxic T cells. On the surface of this cancer cell, they are detecting, using their T cell receptor, certain peptides that are being presented by this cancer cell that these T cells recognize as being bad, right? Because they're not a peptide that normally healthy cells would be presenting. Because if it was a peptide that normal healthy cells are presenting, right, then you would not have T cells like this that have passed tolerance training and are allowed to go to work. So cancer then, if you really end up with full-blown cancer, that must mean what? That these transforming, mutating cells have somehow evaded the immune system. So for cancer to develop, it must escape detection or attack by our immune system. Let's look at this here and appreciate how importantly this is tied in with our immune system. In the lab, let's say you're working with a particular strain of mice. So this yellow mouse here is a genetically inbred strain. So all yellow mice are the exact same genetic background. And we will just call them MHCA, right? So in other words, they all have the exact same set of HLA polymorphisms, so that all of their MHC proteins on the surfaces of the cells are this MHCA type, right? And let's say that this mouse develops a tumor. If scientists isolate the tumor cells or the cancer cells from this cancerous tumor, so that's what these red dots are here, and you go ahead and you inject these cells, these tumor cells, into another genetically identical MHCA mouse, what they find is that these tumor cells will continue to grow in the new mouse and eventually form a malignant tumor as well, right? But if you take those same tumor cells that came from the yellow MHCA mouse and you inject them into a blue mouse, so this is a mouse with a different genetic background, we'll call it MHCB, right? What you find is that the tumor cells are rejected. The animal lives. Why? Because the immune system of the MHCB mice, right, it has the ability to recognize the MHCA polymorphisms on the MHC proteins as being something foreign. So the tumor cells, even though they might be very aggressive tumor cells in the yellow mouse, the very fact that they have different MHC proteins on them, right away the immune system of the blue mouse sees that, right, and is able to kill these mice, not the mice, kill the cancer cells. This is why cancers are not contagious, right? If you accidentally get somebody else's cancer cells into your body, either through breathing it in or who knows what other way. Okay, I don't even want to think about that. Don't worry, your immune system, because you are genetically different than theirs, this is just like tissue graft transplantation, right? If you believe that there's very little chance that tissue taken from another person would be able to actually grow in your body without being rejected, right, then likewise, tumor cells, it's the same thing, right? If it comes from a different genetic background, that means those tumor cells over time found a way to get around that person's specific genetic background of immunity, immune system, right? But once you plop those cells into your body, you're fine because your immune system will detect them as foreign right away. Okay. So let's think about this. Implies that not only MHC proteins, but other things that are presented by the cancer cell might be useful as signs that your immune system can detect that something is bad with this cell. Let's imagine this cell over here. This is a totally normal, healthy cell on the top, right? And it has, let's say, it uses certain genes in the genome that make it this particular type of cell. So let's say this is a muscle cell, for example, right? And so the green protein, the purple protein, and the blue protein come from the green, purple, and blue genes, right? And these are proteins that are useful for a muscle cell. So as a result, we already learned, right? These proteins are constantly being recycled inside this healthy cell, and bits and pieces of these peptides from these different normal proteins are presented on the outside of the cell as a way of your immune system being able to see that everything is good inside of this cell, right? Because your immune system would have been self-tolerance trained not to have things like antibodies, or in this case, T cells that recognize these normal peptide signals. Let's say you start getting mutations. In one scenario, the gene that codes for the blue protein gets a mutation. So as a result, the blue protein now looks a little bit different, right? Part of it is a different shape. As a result, when it gets recycled and processed, the little blue peptide piece that has this different shape on it, which just means what? Different amino acid sequence here, right? This cell, which is a tumor cell that's presenting now this mutant peptide, that could be a signal, right? That your immune system would potentially recognize as being foreign, something different. Those cases, we say that there are antigens being presented that are tumor specific, right? Only this kind of tumor would present this. Other scenarios are, for example, certain genes that these fully differentiated cells normally should not be expressing. So for example, let's say there's a red gene that is an early developmental embryonic gene, right? It's a gene that was last used when you were just a developing embryo in your mom's uterus, right? And long ago, the cells in your body stopped using that gene. Right? But what if a mutation reawakens the expression of that gene? So this tumor cell is now improperly making this red protein, right? So as a result, red protein peptides get presented again, right? This is a situation where it's actually a normal human protein peptide, right? There's no mutation in the peptide itself, but the fact that cells are improperly showing it could be a sign, right? Likewise, let's say this perfectly normal cell that normally expresses the green protein, there's a mutation that causes it to express way more of this green protein than it should. So much more of the green peptides are being expressed on the outside of this cell than it should. Again, right, it's still the normal peptide form of this protein, right? But there's just way too much of it now. So these two examples are what we would call tumor-associated antigens, not something that's unique or specific to a particular tumor type, right? But just different ways of being able to demonstrate that there's something wrong with normal protein expression inside. So this table here, again, just to help you appreciate, are different examples of different peptides, right, that are normal proteins, but mutations in them cause things to go wrong. These are the diseases that they're associated with, things like melanoma, lung cancer, head and neck carcinomas, bladder cancer, leukemia. This is demonstrating that the presentation of peptides from these different mutant proteins, right, would be preferentially displayed by different types of HLA proteins. right? So different HLA-As or Bs or Ds are better at presenting these types of peptides. This is the cool part of the table here, right? This is the actual peptide sequence that very often reflects the mutation that has happened, right? So the amino acid sequence of this peptide has changed, and there should not be, for example, an E here, right? Or there should not be a G here. So there should not be like an aspartate or a glycine here, right? So these are typical peptides that can be detected as being foreign. These peptides come, of course, from the proteasome inside of the cells, processing and recycling these peptides. What this cartoon is showing is that oftentimes, if you have certain proteins that have a mutation in them and the amino acid sequence has been altered, when these proteins are fed into the recycling bin, the proteasome, and they're chopped up into small pieces, a lot of times, these small little pieces of peptides might just sort of have a tendency to stick to each other, to have slightly larger versions of them. But if all of the sequences are normal, then all those different combinations of small peptides coming together, your body would also recognize as being normal, sort of side products of this, right? But in this case, if you have a certain mutation, maybe like in this red region of this peptide, right, that might cause it to join up in a different way with other spliced peptides, and that itself could be presented as being a different peptide by something like a melanoma cell that your CD8 cytotoxic T cells might recognize. Okay. Improperly processed peptides, though, can also serve as a way for the cancer cell to hide from detection, right? And you're like, why, right? If it's wrong or misfolded, wouldn't that actually be a sign that there's something going on and that cells should be able to, of the immune system should recognize that? Well, let's look at this example. MIC or M1C is a protein that's one of the genes expressed in your major histocompatibility complex group of class 1 and class 2 genes. There's sort of a middle region that has some other genes that we didn't really talk about. M1C is one of these. This gene gets expressed when the cell is stressed out or undergoing, some sort of damage. So our immune systems have evolved to detect that if cells are expressing this M1C on their surface, right, then you will have natural killer cells as well as gamma delta T cells that are on the lookout for these types of well-established signals that there's something wrong with the cell, right? So the natural killer cell or the gamma delta T cell, through receptors like NKG2D on their surface, right, will see the M1C on these damaged cells, and they'll eliminate these cells. What if, as part of the development of cancer, this M1C gene itself gets a mutation? So what we often see is that cancer cells, by chance, have gotten a mutant form of this M1C that instead of the protein remaining fixed to the surface of the cell, this pair of scissors show that this mutant form of the protein floats away from the cell. And what happens is because it floats away from the cell, it ends up binding up all of those NKG2D receptors on the surface of something like this blue gamma delta T cell. And as a result, the gamma delta T cell has no idea, right, where this is coming from. And so these cancer cells then have protection through this mutation, and these cancer cells are left alone by our immune system and are allowed to proliferate. Okay, last slide for today, because I know I'm running over time. Sorry about that. I have one more slide after this, but I'll leave it to Monday's lecture. Another way that mutations can help cancer cells hide from the immune system is consider this. So much of what we talk about, how our immune system monitors or checks up on the status of a normal cell that something like a lymphocyte bumps into, is we count on that normal cell to constantly present what's happening inside of it, right? And those cells present things on MHC proteins. If a cell doesn't present things, then in a way, it's like invisible to the immune cells that are coming to visit it. So not surprisingly, if those MHC platforms are coded for by our HLA genes, right, the ones that are part of our MHC complex, what if you ended up with mutations in those genes? So that's what's happening here. Over time, cancer cells may end up with mutations that shut down the expression of normal functional MHC platforms on their surface. So as you see here, in this mix of cells, they're staining for the presence of MHC on these cells. And so if the cells have MHC1 on their surface, they'll stain brown, right? So the scary thing is, this is a cross-section of a cancerous tumor, right? All the cells in here should be staining brown, right? Because they all should be expressing MHC1. But notice, right, many of them have accrued or gained mutations that have actually shut down their expression of MHC1 on their surface. So these cells that aren't brown, that are just kind of like this pale blue, in many ways, these cells are now invisible, right, to patrolling lymphocytes. Okay. All right, let's go ahead and stop there today.